Good morning, students. We are going to continue chapter 6 of NCERT, that is Molecular Basis of Inheritance. This module, students, is really very important from examination point of view and from understanding point of view. As you know, we have already discussed about DNA replication. So the next thing come in our way is the transmission of information from DNA into the cytosol or cytoplasm. How this is being carried out, this module is totally devoted for that process, how the information transmit from the nucleus to the cytoplasm, and that specific process is referred as transcription. As you can see in the animation, there is a DNA strand and some synthesis is going on, particularly on single strand of the DNA. The other strand is totally unaffected. So this particular strand is very very important. We are going to discuss about whole of the process in breadth, length and breadth. So let's continue. Topics which are being covered in this tutorial are transcription, genetic code and mutation. Now, student, first of all, tell me who proposed central dogma of molecular biology. Yes, correct. That is Francis Crick. And what central dogma actually says is that DNA never ever goes outside of the nucleus. Its main aim is to transmit information by the process of RNA, and RNA is utilized for the formation of protein, for which whole of the genome is totally devoted for. So the main aim is the formation of protein, and that information reside under that of the DNA, and the transmission of information or the message from the DNA comes along with that of the RNA, and that RNA ultimately guides the synthesis of protein. As you can see in this particular diagram, DNA never ever comes outside of the nucleus, and DNA transmit information in the form of RNA as a message, that is referred as messenger RNA, and that messenger RNA uh, going to transmit information for the process of protein formation. Okay, so this is very very critical thing you need to understand first. Now comes to the transcription. Transcription simply refers to the copying of genetic information only from single strand of the DNA, not both of the strand of the DNA going to participate in this process. Only one strand of the DNA is going to participate in the formation of RNA, and that process is referred as transcription. Okay, so key points for the process of transcription are: the process of copying genetic information from one strand of the DNA into the RNA is referred as transcription. And the principle of complementarity that applicable into that of the DNA replication is also applicable in this process. As you can see, only adenosine is going to pair with that of the uracil. As you know, uracil is a type of nitrogenous base exclusive for that of the RNA instead of thymine. Okay, so it is almost similar to the process of replication, but there is a quite difference because here not DNA is going to be synthesized, but RNA is going to be synthesized by following the principle of complementarity. Okay, adenosine is going to base or going to form a base pair with that of the uracil and not with that of the thymine. Unlike replication, only a single strand fragment of the DNA is going to participate. And let's discuss further why both the strands are not copied during transcription, why both strands do not going to participate for the process of transcription. As you know, both strength if both strands are going to act as a template and going to form RNA, then these RNA going to form different proteins. Okay, they are going to form different protein. So this will provide lot of complexity in the transmission of genetic information. Okay, so single strand is going to form two different protein, and this will complicate whole of the process of the transmission of genetic information. Okay, so the next complexity when both strands are going to participate in the transcription, as you know, the two RNA which are being formed by the process of transcription will be complementary to each other. Okay, they will form base pair along with themselves, and what will happen to the formation of protein, that will never ever take place. Okay, to avoid both these complexity, only single strand of the DNA going to participate in the process of transcription. These 2 points you need to learn by your heart. Okay, now the next thing, what is transcription unit? It comprises of promoter, structural gene, and terminator. These three units are there in transcription unit. Okay, promoter, it is the binding site of RNA polymerase. As you know that polymerase, whenever the term polymerase comes to your mind, it always going to synthesize the strand in 5 dash to 3 dash direction. Okay, or 5 prime to 3 prime direction. And this particular RNA polymerase is totally dependent on the DNA for the synthesis of RNA. So this is sometimes referred as DNA dependent RNA polymerase. Okay, the complete name of that particular polymerase is DNA dependent because it is completely dependent on the DNA for the synthesis of RNA. So that's why its full name is DNA dependent RNA polymerase. And promoter site is that site where this DNA dependent RNA polymerase is going to bind and start the process of RNA formation. Okay, so next thing is structural gene. It codes for enzyme or protein for structural function. That what's the ultimate aim of the DNA. Next thing is the terminator. It is a region where transcription is going to be end. Okay, and the next 3 points are very very important. You need to understand whole through. Okay, so the polymerase which is responsible for the process of transcription is DNA dependent RNA polymerase. Helps in the DNA replication by catalyzing the polymerization in only one direction, that is 5 prime to 3 prime or 5 dash to 3 dash. As I have told you that polymerase always work in particular direction, that is 5 prime to 3 prime. Okay, DNA strand that has a polarity that is 3 prime to 5 prime acts as a template. So it's going to provide the base for the formation of RNA, and it is referred as template strand or antisense strand. I will tell you later why it is referred as antisense strand. The next thing, the strand which does not get transcripted, which remain unaffected or which do not participate in the process of transcription, is referred as coding strand. And the other name of the coding strand is sense strand, and having its polarity from 5 prime to 3 prime. Now, the term that is coding, which do not code for anything, but it is referred as a coding strand. Why it is so? Because its sequence is same as that of the RNA going to be formed. Okay, so we will discuss one example and you will have the clear cut understanding why this whole nomenclature is like that only. When the strand is not coding for something and it is referred as coding strand, it is totally misnoming term. So why we are using it, I will tell you with one of the example. But first of all, you need to understand 3 prime and 5 prime is going to be your template strand, and it is also referred as antisense strand, and the DNA strand which is from 5 prime to 3 prime is referred as coding strand or sense strand. Okay, so here you can see whole of the transcription unit. Here is the promoter and here is the terminator and you can see structural gene is being sandwiched between the two, that is promoter and terminator region. Promoter is that site where actually DNA dependent RNA polymerase going to bind and form the complete RNA sequence, and terminator is that site where the transcription is going to be stopped. Okay, so here you can see the template strand and coding strand. Template strand is also referred as antisense strand and coding strand is referred as sense strand. Okay, so this type of clarity should be there in your mind before starting whole of the process. It's my humble request to you, please draw this diagram to have clear cut understanding about different parts. Okay, so here the promoter is located towards 5 prime and that is upstream of the structural gene. You can see the both nomenclature, that is promoter and terminator region, is according to the coding strand. Okay, according to the coding strand. The polarity of the coding strand is going to determine the site, that is promoter as well as terminator. Okay, the promoter is located towards 5 prime and upstream of the structural gene of the coding strand. This is very very important point. The nomenclature about promoter and terminator is according to the coding strand and provides a binding site for RNA polymerase. The sequence of DNA located towards 3 prime and that is downstream of the coding strand where the process of transcription will stop, ultimately it is referred as terminator region. Okay, this much should be clear in your mind, that the nomenclature about the promoter and terminator region is according to the coding strand. Promoter region will lie in the upstream, that is at the 5 prime end, and terminator region will lie at the downstream of the coding strand, that is at the 3 prime and that is terminator region. Okay, now the nomenclature, why it is so, you will get answer from this question. As you can see, a hypothetical sequence is there, and here is the coding strand and here is the template strand. Now you have to tell the sequence of RNA which is going to be transcribed from the above DNA. You can see that transcription is going to happen at 3 prime to 5 prime strand, because DNA dependent RNA polymer is going to work only in one direction, that is 5 prime to 3 prime. Okay, so this is your template strand, and A is going to pair with U and T is going to pair with A, C, G with C and C with G, and so on. You can find out whole of the strand. Now compare that particular strand with this strand. You will get the exact replica of it, except at the place of thymidine there will be uracil. Okay, so this much conceptual clarity should be there. The mRNA which is going to be formed from the template strand will have the exact similar base pair as that of the coding strand. So that's why this is referred as coding strand, because mRNA sequence will be similar to the coding strand, not with that of the template strand. So this sequence is referred as coding strand, and it is also referred as sense strand. Okay, so now you should have clear cut understanding why the sequence which is not coding for anything is referred as coding strand, because mRNA which is being derived from the strand of the DNA will have the same sequence as that of the coding strand. Okay, so this much should be clear in your mind first. Now the next thing, what is the difference between template strand and coding strand? Template strand having the this is that kind of a strand of DNA which takes part in transcription, and coding strand do not take part in transcription. Okay, the polarity of template strand is 3 prime to 5 prime, and the polarity is 5 prime to 3 prime in case of coding strand. Nucleotide sequence are complementary to each other. The nucleotide sequence is same as one present in the mRNA, as I've told you before, except the presence of thymidine is instead of uracil. Okay, so this much should be clear in your mind. And template strand is referred as antisense strand, and coding strand is referred as sense strand. Okay, the type of question which actually come from this content is like this type of question. One strand of the DNA will be there, and you can see according to the 5 prime and 3 prime region you can demarcate. A is your what, that is promoter sequence, and T will be your terminator region, and X will be structural gene which is actually being transcribed. Okay, so this type of question actually comes in the examination, and this has been asked in 2019. And you can compare this diagram with the previously studied diagram of us from the NCRT, as we have discussed the structural unit component. So you can go through and mark out all of the X, Y, and A. This question has been asked in the year of 2019 CBSE board examination. And now comes to the transcription unit and the gene. As you know, it is very important to define gene. And in case of eukaryote and prokaryote, it is very difficult to define gene in terms of DNA sequence. So we generally describe cistron. The segment of DNA coding for a polypeptide is referred as cistron, and it this cistron is very different in prokaryote and eukaryote. As you can see, the single strand of the DNA, that is single part, single segment of the DNA, is going to form mRNA. That mRNA is responsible for formation of multiple protein. But in case of eukaryotes, we have different system. Here you can see the mRNA which is formed from structural gene is going to form only single protein. Here you can see multiple protein and only single protein in case of eukaryotes. So this is very important thing you need to keep in your mind. That's why prokaryotes, due to formation of multiple protein from the single mRNA, they are referred as polycystronic, and eukaryotes are referred as monocystronic because their single mRNA is responsible for formation of one protein only. Okay, so eukaryotes are referred as monocystronic and prokaryotes are referred as polycystronic. Okay, now comes to the definition of polycystronic and monocystronic. In eukaryotes, the transcription unit processes a structural gene specify only for a single polypeptide. As we have seen, it is responsible for the formation of only one polypeptide or the protein. Thus, it is called monocystronic. And in case of polycystronic, the transcription unit processing the structural gene for many polypeptide, and which is a part of single metabolic pathway, that is referred as polycystronic. And one more important thing, the structural gene of eukaryotes have so many intervening sequences which do not participate in formation of polypeptide at all. They are just going to be eliminated from the mRNA, final processed mRNA. So these are useless sequences being present along that of the sequences which are going to code for the polypeptide. As you can see here, the DNA is having the gray or a bluish gray thing. These bluish gray spaces are actually introns. Just simply remember introns as a intervening sequences. They are the sequences which interrupt exons at particular sites. Okay, so you can see these are the exons. These exons are actually going to participate in the formation of polypeptide, and in final process mRNA, only the orangish sequences, these are the exons, are there, and intervening sequences are going to be eliminated altogether. So this is very very important aspect. Exons are going to participate in the formation of polypeptide, and introns are going to be eliminated from the mRNA. They are not present in the finally processed mRNA, which is responsible for the formation of polypeptide or the protein or enzyme, whatever. But you need to keep and understand what introns and exons are. Okay, here this is very very important aspect related to that of the eukaryote. Now comes to the transcription in prokaryotes. We are going to discuss transcription both in prokaryotes and eukaryotes. Firstly, we are going to discuss transcription in prokaryotes. In prokaryotes, the structural gene is polycystronic and continuous. You can see that prokaryotes, the structural gene is going to form RNA, which is responsible for the formation of multiple polypeptide. That's why they are referred as polycystronic. Okay, and the sequence is quite continuous. There is no intervening sequences or no introns or any sort of interruption. In bacteria, the transcription of all the three types of RNA, that is mRNA, tRNA, and rRNA. As you know, RNA is of three types, that is mRNA, tRNA, and rRNA, is catalyzed by the single DNA dependent enzyme, that is DNA dependent RNA polymerase. E. coli bacteria also have some cofactors being associated with the RNA polymerase. These are beta dash, alpha dash, and omega, along with sigma factor to catalyze the process. This is very very important aspect when we talk about transcription, specifically in E. coli. Here, DNA dependent RNA polymerase has cofactors. These are six in number, that is beta dash, alpha dash, omega, and sigma factors to catalyze the process. The transcription in prokaryotes is being completed in three steps. These are initiation, elongation, and termination. Sigma factor recognizes the start signal and promoter region on the DNA, that is towards 5' end, which the along with RNA polymerase binds to the promoter to initiate whole of the process. So sigma factor is that kind of factor which is going to demarcate the site for the promoter and ultimately leads to the binding of DNA dependent RNA polymerase. Next step is elongation. The RNA polymerase, after initiation of RNA transcription, loses the sigma factor but continues to polymerize of ribonucleotides to form RNA. Next thing is termination. Once the RNA polymerase reaches to the terminal site region of the DNA, the RNA polymerase is separated from the DNA RNA hybrid. As a result of RNA nascent is going to be separated out from the DNA. This process is governed by one factor that is referred as rho factor. So starting or initiation factor is the sigma factor and termination factor is the rho factor. Okay, these are the cofactors which are actually associated with the enzymes. Now, in prokaryotes, mRNA does not require any sort of processing, so the process transcription and translation can occur simultaneously. Okay, so both transcription and translation occur in the cytosol. It can be said that transcription and translation are coupled together because they go simultaneously side by side. If RNA is going to be synthesized from the DNA, and in the similar with the continuation, the RNA is going to form the polypeptide as well. Now you can see Here, this is a DNA sequence, and sigma factor is going to recognize the promoter region and bind itself there, and it say hello to DNA dependent RNA polymerase, and thereafter it leaves. It has no role, and DNA dependent RNA polymerase is going to polymerize whole of the ribonucleotide to form polyribonucleotide sequence, that is RNA. And if it reaches to the terminator, another factor is going to bind with the RNA polymerase, so that the RNA, the RNA which is being formed from that of the DNA sequence, is going to detach itself from the DNA RNA complex. Okay, here you can see in the animation as well. Here the structural gene is there. Okay, structural gene is there, and messenger RNA is going to be formed. And how it is going to be formed? Because of the factors which recognize the site for the initiation or the promoter site, that is sigma factor, and ultimately DNA dependent RNA polymerase is going to form RNA. Okay, going to form RNA, and terminal site is going to be recognized with the help of factor, that is rho factor. Okay, you need to remember both of the factor, that is sigma factor and rho factor. Now comes to the transcription in eukaryotes. Eukaryotes are monosystronic. The process of transcription is similar to that of the prokaryotes, but there is a much more complexity. I will tell you how. As you know, introns or intervening sequences are there along with that of the exons. So these need to be removed in the further process of the RNA. And one more complexity is there, because there are multiple types of RNA polymerases. These are actually three types: RNA polymerase 1, 2, and 3. RNA polymerase transcribe rRNA, that is ribosomal RNA, 28 Severberg unit, 18 Severberg unit, 5.8 Severberg unit. And RNA polymerase 2 transcribe the precursor of the mRNA called heterogeneous nuclear RNA, hnRNA. You need to remember the full form of hnRNA. It has been asked in the year of 2013, I remember. Uh, there they just asked for the expansion of hnRNA, that is heterogeneous nuclear RNA. And third type of RNA polymerase is responsible for the transcription of tRNA, 5S RNA, and snRNA. snRNA are referred as small nuclear RNA. So there are different types of RNA polymerase for the transcription of different sort of rRNA, hnRNA, snRNA. So you need to remember what kind of polymerase actually transcribe what kind of sequence. Okay, this is very very important from the examination point of view. A straightforward question can be asked from this content. Now comes to the post transcriptional modification. Two complexities we have discussed, that one thing is there is intervening sequences. Second complexity, there are multiple sort of RNA polymerase, that is RNA polymerase 1, 2, and 3. And another modification happens to the mRNA, that is capping and tailing. Okay, capping and tailing refers to the process of addition some of the sequences. Capping refers to the methyl guanosine triphosphate at the 5' end of the hnRNA, and tailing refers to the addition of multiple adenosine bases, around 200 to 300 number. They are going to be added on. Okay, and splicing refers to the removal of introns or intervening sequences uh, from that of the RNA, so it becomes mature mRNA. Okay, Here you can see exon, intron, exon, intron, exon, intron. So these introns or intervening sequences has no role in the formation of the polypeptide. They need to be removed. And how they are going to be removed? With the help of spliceosomes. A special type of enzymes are there to remove these intervening sequences. And here you can see capping and tailing is going to be take place. Methyl guanosine triphosphate is going to be added at the 5 prime end, and multiple poly A tail is going to be added, that is adenylated bases are going to be added. And here you can see spliceosomes are going to excise or remove all sort of introns from that of the mature mRNA. And here also you can see the complexity in the eukaryotic mRNA. Here introns or intervening sequences are going to be removed altogether. Capping, that is 5-methyl guanosine triphosphate is going to be added, and polyadenylation is going to be take place, that is a poly A tail around 200 to 300 base pairs are going to be added there, uh, added at the 3 prime, and this is referred as poly A tail. Okay, so why this capping and tailing actually happens? We know the reason how why introns are going to be removed, because they are not going to participate ultimately in the formation of polypeptide. But why capping and tailing is going to be take place? As you know, this mRNA is going into very harsh environment of the cytosol. So many enzymes are there. So these type of protection, that is capping and tailing, protect the internal sequence, that is the sequence which is responsible for the formation of polypeptide. This is type of some sort of protection at both the end, that is 5 prime and 3 prime end is there to protect the nascent mRNA. Okay, so how DNA replication is different from transcription? This is very very important question and can be asked into the examination. So this is the particular format in which you have to answer. First of all, the function of DNA replication is to make the copy of the DNA or synthesize the identical copy of the DNA. The next thing, that is a function of transcription, that is to make RNA from the DNA. And enzymes involved in the replication are helicase, polymerase, and ligase. But the enzymes which are responsible for the transcription are DNA helicase and RNA polymerase. Don't worry about the helicase. As you know, our DNA is helical, double helical in fact. So this helicase is going to unwind the DNA to form a single stretch or the regular stretch. Okay, so it is going to remove all the helical nature of the DNA. So this enzyme is common both for the replication and transcription. The nucleotide sequence are deoxythymidine monophosphate, deoxycytosine monophosphate, deoxyadenosine monophosphate, and deoxyguanosine monophosphate. And here you can see UMP, CMP, AMP, GMP is going to participate. T, C, M, G, these are the TCAG are actually the base pair, and here UCAG are the base pair. DNA replication takes place in the S phase, and transcription takes place at the G1 and G2 phases. It occurs for both leading and lagging strand. We have discussed in the previous module, but it talks about that is leading and lagging strand and preparation for the cell division. But when transcription is actually happening, it occurs at G1 and G2 phase along one template DNA strand, preparation for the gene expression. Bonding, copied DNA bonded to the template DNA, but here copied RNA is going to separate from the DNA to go into the cytosol. DNA replication requires primer, but gene transcription do not require any sort of primer. Products which are going to be formed by the replication are DNA, daughter strands, and stay within the nucleolus, and not going to be degrade. But here the processing of the RNA takes place. As we know, there is a removal of introns takes place. So mRNA is going to ultimately destined to go inside that of the cytosol for the formation of polypeptide and degraded after doing all sort of work. Copying, that is entire genome is going to copy, but here in transcription only individual genes are going to be transcripted. Unwinding of the DNA usually takes place whole of the DNA, entire DNA. But here the sequence which is going to form RNA is going to unwind only. Here you can see post-copying process, that is DNA ligase to join all of the Okazaki fragments. Here RNA editing takes place, introns, deleted exons are going to be spliced off. Okay, so this type of processing actually happens into the transcription. Now comes to the genetic code. This is very important aspect. We are quite aware what kind of nitrogenous base pairs are there, but what sequence actually determine what kind of amino acid to be inculcate for the formation of polypeptide. This is going to be very challenging task because we have four nitrogenous base pair, and how their sequence is going to determine what kind of amino acid is going to participate in the formation of polypeptide. This is very crucial question which need to be answered, and it almost took a decade to being deciphered. First of all, first physicists participate in the decipheration of the genetic code, that is George Gamow. As he was a physicist, he had a great knowledge about the mathematics, so he just simplified whole of the thing. As you know, 20 amino acids are there and 4 base pairs are there, so all in all 64, 4 is to the power 3 is going to achieve the target of 20 amino acid. Why it is so? If we only took 4 bases, it's not going to generate 20 amino acid sequences. So we take another 4 one, because we have 4 base pairs, it makes 16, but it is not up lesser than that of the number of amino acid required for the formation of polypeptide, that is 20 amino acid. So the next alternative is to multiply by 4 again. It will become 64. So codon must be there in the form of triplet. Next breakthrough was came from Hargobind Singh Khurana. He was pioneer for the artificial synthesis of DNA, RNA, both in the laboratories. So he developed chemical methods for synthesizing RNA molecule with defined base sequence and developed the genetic code for the same. Marshall Nierenberg artificially synthesized protein to understand nature of codons, because this is very important to synthesize protein in vitro medium to get to know how the information is actually being transmitted. And the final thing was done by Severo Shaw, demonstrated that polynucleotide phosphorylase also helped in polymerizing RNA with defined sequence in the template independent manner, that is enzymatic RNA synthesis. He derived that how RNA synthesis is actually completed with the help of enzymatic activity. You can see in the year of 1968, along with Holle, Hargobind Singh Khurana and Marshall Nierenberg received Nobel Prize in Physiology or Medicine. This is the dedicated photo of Hargobind Singh Khurana, because he was of Indian origin, and we need to read more about him. Here, this is the beautiful representation of the codons deciphered by Hargobind Singh Khurana actually. Here you can see 4 nucleotide are there, and again 4 nucleotide, and you can see u is going to multiply with u, and here again u is there, so u c, u a, and u g. You have to follow the triangle, u multiply by u, and then this u need to be for that of the third position, as we have discussed the George Gamow decipheration. He said that 4 into 4 into 4 is going to make ultimately the 20 amino acids. So this how we need to place for the formation of a particular codon which is responsible for particular amino acid. Here you can see UUU is responsible for the phenylalanine, and here serine is derived from this UCU. You need not to learn whole of the table, but some key points you need to remember. These are the of the stop codon. Here UAA, UAG, and UGA are the stop codons, and AUG is the initiator codon. AUG methionine is the initiator codon. Some key facts you need to remember, otherwise no need to remember whole of the table, but you should have clear cut idea how it is being derived. There are silent features of genetic code, and any question can come from these 6 points. Okay, so you need to learn these features by your heart. The codons are triplet. Out of 64 codons, 61 code for 20 amino acids and 3 for uh, are of none of use. These are the stop codons. As I've told you before, UA, UGA, UAG are the stop codons. And one codon codes for only one particular amino acid, hence the code is unambiguous and specific. Some amino acids are coded by more than one codon, hence they are degenerate. The codon is read in mRNA in a contiguous fashion. There are no punctuation in reading of the codon. They are comma-less actually. The code is nearly universally applicable for all of the species present on the Mother Earth. For example, from bacteria to human, UUU is code for the phenylalanine, and some exceptions to this rule has been found in mitochondrial codon. That is very very important. Mitochondrial codon is quite different. As you know, mitochondria is that organelle which has its own DNA content, but the codon sequence is not follow the table which we have discussed. The codons are different, and they are going to produce different amino acids. Okay, and especially in the case of protozoans as well. AUG has dual function. It code for methionine as well as act as an initiator codon. Now comes to the gene mutation. We have discussed mutation both in terms of genes and chromosomal abbreviation in chapter 5th, but now we have to discuss about its type. We are focusing only on the gene mutation, not on the chromosome mutation in this chapter. Point mutation and frame shift mutation. Point mutation is that kind of mutation where only single base pair is being changed with someone else. Like in case of transition and transversion, in case of transition, pyrimidine replaces pyrimidine and purine is going to replace the purine. But in transversion, purine actually replaces pyrimidine and vice versa. Frame shift mutation talks about insertion and deletion. It talks about addition of some base pair as well as deletion of some base pair. Here, the two types of mutations are there. Point mutation, it is a mutation in a single base pair which is replaced by another base pair. For example, in case of sickle cell anemia, we have discussed that point mutation took place in a beta globulin chain, and the resultant amino acid is different, that is glutamate to valine. And now comes to that of the frame shift mutation. It is change in the reading frame because of the insertion or deletion of the base pair. Insertion is the addition of the base pair in the DNA sequence. Okay, insertion can be of a multiple DNA sequence, nucleotide can take place, and it change whole of the frame of the reading for the codon. Deletion refers to the removal of one or more nucleotide from the DNA segment. It can happens to that of the three at a time or multiple base pair. And here you can see a normal sequence. If insertion took place here, that is C GAT. Here C has been added, whole of the frame has been changed. And deletion, here the deletion took place and whole of the frame has been changed. So these type of conceptual clarity should be there in mind before going further for the text. That's all for today. Thank you.